Hello everyone, today's our video is going to be mainly on lesson 28 all the way to lesson 32. The majority of today's review is going to be focused on the reproductive systems as well as mitosis, meiosis, and human development from one cell all the way to being delivered as a normal baby. For chapters that are about homeostasis, human systems, please refer to the previous test review, the test 4 review or test 5 review. I will put the link down later. If you wanted to briefly refresh your memory on homeostasis and human systems and cell types. So first thing that you need to remember for this coming test is the name of the structure as well as its functions. For male, the reproductive system is the penis, the testis, epididymis, urethra, scrotum, the gland, vas deferens, and that's it. Remember the functions of each part of the structure. Urethra is part of the urinary system, so we're just going to ignore this as of right now. And seminal vesicle is not covered in our syllabus, so we're not going to talk about it. So the organ that produce the sperm cells, the sex cells of the male reproductive system is the testis. So testis produce sperm cells. And the sperm cells is going to be stored in epididymis. So epididymis is like a storage, so it stores mature sperm cells. When sperm cells need to be released, it will be released through the tube called vas deferens. So vas deferens is a tube that channel the sperm cells out of the testis, right, into the female reproductive systems. And when it passes through the vas deferens, it's going to pass through a gland, the prostate gland. Prostate gland secretes fluid as well as some other hormones that's needed for the sperm cells. So fluid mixed with sperm cells is going to form the solutions, which is called semen. So those fluids are important to nourish the sperm cells because the sperm cells need to move around. They need the nutrients. Next, erectile tissue is the tissue that's surrounding the urethra. So once vas deferens connecting to the tube, right, it will be sharing the tube with the urinary system called urethra. So for male, they urinate and ejaculate through the same tube, which is called urethra. Erectile tissue will be surrounding the urethra. Is the main structure of the penis. So when penis needs to be erected, this tissue will be filled with blood. But it's not the focus of our lesson, so I'm just going to talk about it briefly. Scrotum is basically the skin tissues that holds the testis. Both scrotum, testis are outside of the male's main body, because sperms needs to be stored at a lower body temperature in order to be produced efficiently and effectively. Okay, so remember that scrotum is the location where it holds testis, and it needs to be outside of the main body because it needs a lower body temperature than its main body. Next, we're gonna move on to female reproductive system. Remember the name and the locations of the structure. We're going to start from ovaries. Both male and female have a pair of gametes producing organs. For male, it's called testis, and for female, it's called ovary. So notice that there will always be a pair. So ovary is the location for female to produce their sex cells, which is egg cells. So produce egg cells. So once egg cell has been produced, it will then be released to the oviduct or fallopian tube. So both names are correct. They both refer to the same thing, which is the tube that connects the ovary to the main structure over here, which is uterus. So uterus is, in a layman term, the womb. It is like the big location where female, when they're pregnant, they keep their baby, right? So this is where a little embryo is growing for 10 months in this location, uterus. We're going to talk more later in detail about the structure of the uterus. And then we have endometrium. Basically, it is the uterus wall or the lining of the uterus. This part is very important. It is because this is the location where the embryos will be implanted, right? The embryo, or you can say as the fertilized egg, the zygote. This is where the implantation will happen. Implantation of zygotes will happen at the endometrium, or the lining of the uterus. Cervix is the neck of the uterus. It's the opening for sperm cells to enter into the uterus, or for the menstruate to be discharged out of the uterus. A vagina is the location where penis is inserted into and release the sperm cells so that the sperm cells can enter into the uterus and fertilize the egg at the fallopian tube. So remember where the process happens at different locations. Fallopian tube is where fertilization occurs. Fertilization occurs here. Fertilization is the fusion between the sperm cells and the egg cell. Okay, and once the eggs fertilized, the egg will then move to the endometrium. So this location over here, this location, the endometrium or the wall of uterus will become thicker and thicker in order to prepare for the arrival of the fertilized egg, so that the fertilized egg can then be implanted over there. So implantation happens at the endometrium in the uterus, and then the baby will start growing in this area, which is the uterus. Okay, remember that the functions of vagina, which is to it is the entrance for the penis. It is also the location for baby to come out, and it's also the location where female discharge their menstruates. Okay, so now we have covered female. Next, moving on to the pathway, right? So this is just a brief summary of what I said just now. Sperm is being produced as the testis. When ejaculation happens, so ejaculation, it is the release of sperm cells. You can notice that the sperm cells will follow the blue arrows, right? So sperms will be stored in epididymis first, and then it will be released through the urethra and vas deferens. It will pass through the prostate screen in order to mix with the fluid produced by the prostate gland to form semen, and then it will be released and enter through the cervix. Cervix is the opening of the uterus. Male will always produce millions of sperm cells. They kind of have to, because this is to increase the survival of the sperm cells. Cervix and vagina is acidic, so most of the sperm cell will get killed during the process of entering the female body. Furthermore, the female's body's immune system is going to target the sperm cells as invaders. So because it's foreign cells, they don't recognize that cell, so they're going to attack those sperm cells. Cervix is very small, and usually there is a lining here to prevent the entering of foreign cells or foreign particles. So only a few of the sperm cells will be able to penetrate and enter into the uterus and the fallopian tube. During ovulation, only one out of the two ovaries is going to release an egg cell. But sperm will not be able to know which ovary is producing and releasing the egg cell. So half of the sperm cells will move to the wrong part of the fallopian tube, and another half of them, the survived one of course, will move to the fallopian tube where the egg cell is located at. And eventually one of the lucky sperm cell is going to be able to fertilize the egg. So remember, egg will meet the sperms at the fallopian tube. If it successfully is being fertilized, it will become zygote. So remember that this is very important. Zygote is the fertilized egg. With that, let's talk a little bit more about ovulation. So this is a zoom in picture of the ovary. You can see that this is actually the left ovaries. We didn't really talk much about how to tell whether it's left or right, but it doesn't really matter. Inside the ovary, we have the egg cells being surrounded by follicle. We can ignore the primary follicle and secondary follicle and vesicular follicles. It's not important, but the idea is that egg cells doesn't just pop out of nowhere. egg cells growing together with a structure called follicle. So you notice that this structure over here that's surrounding the egg cells in here, right at the center here, this is the egg cell. Egg cell is being surrounded by follicle tissue. So follicle is going to support and nourish the egg cells. It's going to give nutrients to the egg cells. And later, during the right time, we're going to talk about when is the right time, but every 15 days, every 30 days, my bad, every 30 days, about 30 days, like 28 to 30, every 28 days, an egg cell will be released out of the ovary. The process of releasing egg cell out of the ovary into the fallopian tube is called ovulation, right? There are many names that we need to remember. Ovulation. Ovulates, derived from ovary, right? It's related to females. So ovulate, release the egg cells into the fallopian tube. Once the egg cells being released out of the ovary into the fallopian tube, the leftover follicles here is going to get degenerated into what we call corpus luteum. So corpus luteum is the leftover of the follicle after the ovulation. So this follicle actually has some functions of that. It's actually the main structure of producing the hormones needed to support the growth and thickening and maintaining of the endometrium. Okay, so remember the keyword here. We covered follicle, ovulation, as well as corpus luteum. They're all found in the ovary. Next, we already talked a little bit about sperm cells and egg cells, right? So fertilization is basically the fusions of a sperm cells and an egg cell. So a sperm meets with the egg, and when they combine together, they're going to form a complete cells, right? So their DNA is going to fuse together to make one cell, a complete cell. So a complete cell is later going to be further divided into 2 cells, 4 cells, egg cells, and eventually going to become the million cells that makes what you are. Humans has 46 chromosomes. So it means that every cells in our human body should have 46 chromosomes. They must have 46 chromosomes. But if sperm cells has 46 chromosomes and egg cells 46 chromosomes, and when they fuse together to become a complete cell, they will end up having 92 chromosomes, which is impossible. It's not going to happen to make a human, because human need to have 46, but if sperm has 46 and egg is 46, you're going to have 92. So the idea is that the sperm and egg cells needs to have half the number of chromosomes. They can only have 23 chromosomes. So the big idea is that all cells in your body has 46 chromosomes except 2. And depends on which sex you are. If you're male, your sperm cells only has 23 chromosomes. And if you're female, your egg cell only has 46 chromosomes. So far so good. So the idea is that sex cells or gamete cell only has half of the number of chromosomes of that particular species. So let's say if I assume that Drosophila, so fruit flies has 8 chromosomes for every cells found in the fruit flies. So fruit fly sperm cells must have only half of its chromosome. So the sperms of fruit flies can only have 4 chromosomes, while the egg cells can only have 4 chromosomes as well. So that when they fuse together, it becomes 8, which is the number of chromosomes of the fruit flies. Most of the cells in our body reproduce through the process of mitosis, which is just the reproducing of cells that are genetically identical to each other. But sperms and egg cells are not genetically identical to the cells in our body because their chromosomes number are being half. So they are being made through the process of meiosis. Okay, so know the difference. The idea is that all our cells here need to have the same number of chromosomes. So they can only use the process called mitosis so that the number of chromosomes will stay the same. If you start at 2n, you always end up with 2n. It's cloning. Mitosis will produce cells that are genetically identical. They are the same. You can notice that the colors are the same. You have dark red, middle red, dark blue, light blue, and they'll always end up being dark red, light red, dark red, not so dark red, light blue, dark blue, right? Their cells, the chromosomes color is the same. Meiosis is going to halve the number of chromosomes. So if I start with 2n, I will always end up with cells that are only n, half the number of chromosomes. They will be genetically different from each other. You can see that the cells here, they have different colors of chromosomes. That tells us that the chromosomes are now genetically different from each other. So know the difference. Mitosis make all the cells in our body except gametes, and meiosis makes only gamete cells. So here I make a table for you to know the difference between mitosis and meiosis. Mitosis is a process of... I noticed something. Yes, no, sorry, my bad, I took it back. I thought I typed the wrong word, actually it's correct. Mitosis is a process of asexual reproduction, no sex involved. A means no sex involved, right? The cells are going to divide into two, producing a replica, equal number of chromosomes, resulting a diploid cell, a diploid cell. So basically, diploid means you have the double number of chromosomes. Haploid means you have half the number of chromosomes, right? So we started as diploid, we end up as diploid. Meiosis is a type of cellular reproduction which the chromosomes will be reduced by half. So if I start with 2n, I will end with n. And the chromosomes will be separated, right? The chromosomes, homologous chromosomes, they get separated in pairs. The paired chromosomes being separated. If you are confused by the idea of homologous chromosomes, just imagine that I put a pair of chromosomes next to each other. So this is a pair of homologous chromosomes. One from father and one from mother. At the end of meiosis, you're going to produce two haploid cells. Haploid means half the number of chromosomes. So mitosis, the function is for growth, repair, and replace cells. Meiosis, the function is to make gamete. Type of reproduction, it is asexually, sexually. They will be genetically identical for mitosis, but meiosis, their DNA will be different for the daughter cells that are being produced. So they're genetically different. Crossing over will not happen in mitosis, but in meiosis it will happen, most probably. It will happen during the process of prophase 1. Okay? So crossing over is basically the exchange of genetic material, basically DNA, exchange of DNA. Pairing of homologous chromosome is not going to happen in mitosis, but it will happen in mitosis it will happen in meiosis, my bad. It will happen in meiosis 1. Okay? For mitosis, each mitosis is going to divide one time. But in meiosis, it will divide 2 times. Meiosis 1 and meiosis 2. At the end of mitosis, you will end up having 2 daughter cells. I will end up making 2 cells. But in meiosis, I will end up having 4 cells. Chromosome number, it will be maintained. If I started with 48 chromosomes, after mitosis, I will end up having 48 chromosomes. SOMES, SOMES. But for meiosis, it will get half. If I started with 48 chromosomes, I will end up with 24 chromosomes. The step for mitosis is prophase, metaphase, of course, we all started with interphase first, right? So interphase, both started with interphase, but then after interphase, it will be prophase, metaphase, anaphase, then telophase, then cytokinesis. For meiosis, it will start with interphase. Then, during the process of meiosis, it will be prophase 1, then metaphase 1, then anaphase 1, then telophase 1. Right after telophase, it will then end. It will then start with prophase 2, then metaphase 2, then anaphase 2, then telophase 2. Then cytokinesis. Okay? Notice that for meiosis, we code the name as 1 or 2. So if you see prophase but there's no 1 or 2, you know that we're referring to mitosis. If I say prophase 2, then you know that it is a part of the process of meiosis. Only meiosis has 1 or 2 as the code for the steps of its process. Both are going to have cytokinesis and both are going to happen something is wrong here. After telophase 1 here, there's going to have cytokinesis. Right, cytokinesis is separation of the cytoplasm, so of course it's going to happen here. And then prophase 2, then after telophase it will be cytokinesis again. Cytokinesis is a separation of the cytoplasm and the formation of new membrane that surrounds the cytoplasm to form 2 cells. So in mitosis, cytokinesis is going to happen after telophase, so it's yes. And for meiosis, it's going to happen after telophase 1 and telophase 2, so the answer is also yes. So 2 times, and for mitosis 1 time. Okay, mitosis is going to create all kinds of cells in our body. We call this kind of cells somatic cells or non-sex cells. In meiosis, it will only produce gamete cells, which are sex cells. And we only have 2 types of sex cells, which is sperm and egg. Okay, here is a simple picture of mitosis: prophase, metaphase, anaphase, telophase. Prophase is the disintegration of the nuclear membrane and chromosomes begin to condense. Next, metaphase, the chromosome is going to line up in the middle, and then spindle fiber is going to form and attach to the centrosomes. Centromere, centromere at the center of the chromosomes. And the anaphase, the chromosomes will get split into 2 sister chromatids. So these are sister chromatids. Sister chromatids will get separated. And then at telophase, nuclear membrane is going to be reformed again. Spindle fiber is going to stop. It's going to disintegrate it. And then you can start seeing this pinching process, right? The cell is going to start pinching. And after that, we're going to have cytokinesis, which is the formation of 2 new cells. They're going to be genetically identical, which means if I have 4 chromosomes here, I'm going to have 4 chromosomes here as well. And the parent cells, the original cells before the splitting, is also going to have 4 chromosomes. So you can see that in this case, before splitting here, right, there should be 8 chromosomes here. So what's going to happen? So why does this happen? How did 4 chromosomes end up having 2 cells that have 4 chromosomes each as well? This happens because DNA gets doubled right before the process of mitosis. So before prophase 1, DNA has to get duplicated, so that there's actually 8 chromosomes in there, but we just couldn't see it. Another thing that we need to pay attention to is how the method of how biologists count the number of chromosomes, right? We count the number of chromosomes by counting the number of centromere, right? Black dots over here, the center of the chromosomes. That dot over there would be the way how we count the number of chromosomes. And in this picture, we see that there are 4 dots over here. So we say this cell has 4 chromosomes. Next, you also need to learn how to count sister chromatids, right? Sister chromatids is basically, you can see that the arms of the chromosomes, right? So in this picture here, you can see that there are actually 8 arms of that chromosomes. It's just that they're being attached to by the centromere at the center. So we say that in this picture, this cell, there are actually 8 sister chromosomes, but they are being attached by the centromere. So 8 sister chromosomes. Chromatids, my bad, sister chromatids. In this picture, the chromosomes being split, right? So we still count the center, we still count the number of chromosomes by the center of the chromosome, which is the centromere. So in this picture, we said this cell has 1, 2, 3, 4, 5, 6, 7, 8. There are 8 chromosomes in this cell. How about the sister chromatids? So we're going to count the arms of each chromosome. So 1, 2, 3, 4, 5, 6, 7, 8. So in this cell, there are actually 8 sister chromatids. Okay? So hopefully you guys have a good picture and a good sense of how to count the number of chromosomes. Okay. Next, we're going to move on to meiosis 1 and meiosis 2. Notice that the clear differences here is the arrangement of homologous chromosomes. You can see that it comes in as a pair of chromosomes in here, right? A pair of chromosomes, we say a pair means 2, right? So the 2 chromosomes are being paired up right at the center. In what phase? Metaphase 1. So this is the major differences here between meiosis and mitosis. in prophase 1, spindle fiber is going to form, nuclear membrane is going to disintegrate it. But you can see, notice here, this very important part here. You can see that the chromosomes are doing what we call the crossing over, which is the exchanging of DNA between the homologous chromosomes. So this is important because exchanging DNA is going to create a variation among the genetic material of each cell. That's how we create diversities and differences between you and your siblings and your parents. And we mentioned the importance of having variations in our genetic material, right? So that if there is like a virus that can kill one person's because of its immune system, because of his or her immune system, other people will not get affected too much because we don't really share the same immune system as that person's. We have different genetic background as the person who just got killed by the virus. So we're safe. Not all of us will get wiped out by one single virus. Next, let's get back to meiosis. Another thing that is different between meiosis and mitosis is the anaphase 1. In anaphase, in mitosis, it is a separation of sister chromatids, but in meiosis, it is the separation of homologous chromosomes. You can see that it is actually the chromosomes, the whole chromosomes being split apart into the cell, right? So anaphase 1, homologous chromosomes are separated. E-A-R-A-T-E-D. Meiosis 2 is very similar to mitosis, right? If you pay attention to the detail here, you can see that if I just cover the name over here, and you look at the picture here, it looks just exactly like mitosis. So prophase 2 is going to be very similar to prophase in mitosis. Metaphase 2 is going to be very similar to metaphase in mitosis. Anaphase 2 is going to be very similar to anaphase in mitosis, right? The key difference here is that in meiosis 2, each cell only has half the number of chromosomes as before, right? You see here, we have 4 chromosomes in these cells, and in mitosis, there's also 4 chromosomes in these cells. And at the end of mitosis, each cell only has 4 chromosomes, right? It still retains the number of chromosomes, but in meiosis 2, it becomes 2 chromosomes. Okay, so hopefully, you notice the differences between meiosis 1, meiosis 2, as well as mitosis. It's quite important to know the differences. The idea is that meiosis 2 starts with half the chromosomes than mitosis. Next is the egg cell and the sperm cells, right? We said that egg and sperm cells are the gamete cells, G-A-M-E-T-E. They are part of the cells in reproductive systems, and their only job is to fertilize each other in order to become a zygote. And the zygote, if implanted correctly and healthily in the endometrium, is going to start growing, and eventually it will become a baby, and it will get delivered, and you know, maybe he will become my student in the future. Well, who knows? Okay, back to the biology. So egg cell and sperm cells are what we call gamete cells or sex cells. They have some similarities. They are both haploid, right? So it means they only contains half the genetic material from the parent cell. Okay, and they are part of the reproductive system. There's some differences between egg cells and sperm cells. I mean, most common one is that the egg cell is a female egg cell. It's a female sex cells, while sperm cell is a male sex cells. Sperm cells are very small, and they are very motive. They move around a lot, and they connect a lot of mitochondria because they need to support the movement. Sperm cells has a very big flagella at the end of the cell, and is being made in huge number, while egg cells is huge as compared to the size of a sperm cells. Is only made one. It's not multi, it's mobile. Just realize, my bad, mobile. Okay, egg cells are made once every month. It's being released once every month. I mean, in female body there are a lot of egg cells. It's just that they're not being released out of the ovary. But every time when ovulation happens, there will only be one egg cell being released into the fallopian tube. One cell is released into the fallopian tube. oviduct. And it is very not mobile. Okay, it doesn't move around very much. Doesn't have a flagella as well, and it's huge, it's brown. Okay, so the release of this egg cells and the whole cycle of this is being regulated by hormones, right? There are different types of hormones in female body that kind of fine-tune this whole cycle process. This cycle is what we call menstruations, right? Menstrual cycle. It's called cycle because it's been repeated every 28 days. I mean, there's some variety in there, and but it's about 28 days. First is the hormones FSH and LH, right? FSH is follicle stimulating hormone. The name itself kind of tells you what it does. It stimulates the production of follicle in the female's ovary. LH is luteinizing hormone. Both hormones are being produced in anterior pituitary, which is part of the brain. It's a gland very close to the brain. It's part of the endocrine systems. So anterior pituitary is going to release these two hormones, FSH and LH. FSH is going to start increase and stimulate the production of follicle, right? So follicle is going to start producing if there is a high level of FSH. Then after 15 days, 13 to 14 days, luteinizing hormone is going to start peaking out. You can see that LH is this pink color over here. Luteinizing is going to peak at day 13 and 14. The peaking of this LH is going to stimulate ovulations. So LH's function is to stimulate the release of egg cells out of the ovary into the fallopian tube. This process is called ovulation. So LH function is basically for ovulations, right? Once ovulations happening, these egg cells, if fertilized, will need to have a thick cell wall to be implanted so that the cells can get the nutrients from the mother. To regulate the endometrium or the uterus lining, we need two other hormones, which is estrogen and progesterone and estrogen. First, let's focus on estrogen, right? Estrogen is going to start stimulating the thickening of the endometrium. So, with the release of estrogen is going to make the endometrium become thicker and thicker as day goes by, right? So this demonstrates the thickness of the endometrium. If I just look at here, right, it's gonna get thicker and thicker. While progesterone is going to maintain the thickness, it's gonna make sure that the endometrium is gonna stay thick, so that the fertilized egg can be implanted at the endometrium successfully. Both of these hormones are going to be produced by the ovary. Okay, so both of these hormones are produced in the ovary to stimulate the increase in thickness of the endometrium, as well as maintaining the thickness of the endometrium. In this case, two things will happen. If the egg is being fertilized, this fertilized egg is going to be implanted at the thickened endometrium, and then growth is going to start happening on that egg cells, that fertilized egg or zygote. This time we can also call it embryo. Okay. If the egg cell is not fertilized, then that egg cell would not get implanted into the endometrium, and all things will go wasted. So this egg cells will need to get discharged, as well as the endometrium. So the thickness of the endometrium will start disintegrated, so it will become thinner and thinner. So endometrium is gonna get thinner. At that time, all the wasted or a disintegrated endometrium needs to get discharged. So the discharge of all these wasted tissues are going to be released through cervix and out of the female body. This whole process is called menstruation, right? Menstruation. So the discharge of the unused or tissues of the endometrium. And this whole process will get repeated again after menstruations. So we have finished talking about menstrual cycle. Next, let's talk about the fertilized egg cells, right? So it's a repeat of what we have covered just now. Ovulation is the release of egg cells out of the ovary. If the egg cell has been fertilized, day 0 is the fertilized. There you can notice the fertilization happens in the fallopian tube. Right after fertilization happens, this fertilized egg or zygote is going to be divided rapidly from 1 cell to 2 cells to 4 cells to 8 cells and then to a lot of cells if it keeps divide and divide and divide. This process takes about 5 to 6 days, and the name of this process is called cleavage. It's the rapid divisions of the cells from 1 cell to many cells. What process is involved? Mitosis. Because it's cloning from 1 cell to 2 cells to 4 cells, all these cells are going to be genetically the same, right? Eventually, this whole cell is going to become a human or an organism, and that organism is going to have the same, is going to have cells with the same genetic material. And we're not going to focus on morula, so you don't have to know what morula is. But eventually, this bunch of cells, right, this conglomerate of many cells is going to start differentiate into an inner layer and an outer layer. This whole clump of cells is what we call blastocyst, right? By the end of rapid divisions, where you have many cells in one clump, it's going to differentiate into inner cell mass at the inside here, the inner region over here, and the outer layer of that cell is what we call trophoblast. In between inner cell mass and trophoblast, there's really just empty space, which is called blastocoil, but we're going to ignore what blastocoil is. So a blastocyst consists of inner cell mass, which eventually is going to become the organism, which is you or the baby. Trophoblast is going to become what's being attached to the mothers, as well as the placenta. Okay? And this blastocyst is going to start getting implanted into the endometrium. If the endometrium is thick enough, right? It's the embedding of that blastocyst into the endometrium. This whole process is called implantation. The embedding of blastocyst into the endometrium for growth of the baby. Okay, so cleavage is mitosis, rapid divisions from zygote of fertilized egg, to blastocyst, which consists of inner cell mass, which is really the stem cells or the embryonic stem cells that forms every cells in the body, as well as the trophoblast, which makes the placenta as well as other important organ parts that is needed by the baby to have a exchange of nutrients and waste with its mother. Okay. So here is a picture of the implantation process, right? Once this blastocyst is being implanted into the endometrium, it's going to start forming different layers or membranes of that blastocyst, such as amnion, chorion, yolk sac, as well as alitonis. And those structures not going to be very important. It's not going to be emphasized a lot in this test. Just remember that you need to form a few structure. First is the placenta, which is made from trophoblasts. Right? Placenta is the important structure for the baby to exchange nutrients, blood, oxygen, as well as releasing waste of the babies to the mothers. And the inner cell mass is going to form some interesting structure, which is the germ layer. Okay, so this process, as here we can see the cleavage is basically the rapid divisions of one cell, the zygote, all the way to many cells and become blastocysts. And the inner cell mass is going to start folding and form 3 germ layers. So the 3 germ layer is ectoderm, mesoderm, and endoderm. The process of this particular process is called gastrulation. Okay, another word here, gastrulation. So gastrulation is the folding of the inner cell mass to form 3 distinct germ layers that eventually becomes all parts of your body: the ectoderm, mesoderm, and endoderm. Ectoderm is going to form the outer layer lining, such as the skin cells, as well as the central nervous system. Endoderm is going to form the tube of your body, so every inner organs that are connected to the tube, such as the digestive tract, as well as your respiratory tract, right? Such as for digestive tract, your esophagus, stomach, and intestines, and everything in between your skin, central system, the central nervous system. Everything that's in between your central nervous system and the digestive tract and the respiratory tract will be formed from mesoderm. So mesoderm is in between, middle. Mesoderm is going to form all the connective tissue in between ectoderm and endoderm. So here is a picture of the implantation. Right at the beginning of the implantation, gastrulation is going to happen. It's going to form ectoderm, mesoderm, and endoderm. The different layers of membrane is going to form that surrounds the embryo. And then eventually placenta is going to form from the trophoblast, and some of the membranes are being formed just now here. Placenta is going to be important for the growth of the baby, because this is the region where baby and mother exchange nutrients, oxygen, and waste in between the mother and the baby. And then after a few months, it's not few months, 10 months later, baby is ready to be born, right? The process of delivering the baby is called laboring, right? The delivery of the baby. This process is a positive feedback. Why is this a positive feedback? Because of a very special hormone called oxytocin. When the baby is ready, the mother is going to release oxytocin. Okay? And oxytocin is a hormone that can stimulate the uterus to contract and stimulate placenta to make other hormones. It's not very important, but it's going to stimulate more contraction of the uterus. So uterus is going to start contracting in order to push the baby out of the uterus through the cervix and vagina. And this particular process is going to cause more oxytocins to happen. So this is a positive feedback, right? The contraction of the uterus is going to send signal to the brain, the pituitary gland, that, hey, you need to make more oxytocin so that it can contract more so that it will be able to push the baby out more. You need to keep pushing and pushing and pushing until the baby is being pushed out of the uterus. So this is a positive feedback. And with that, we finish the whole reproductive system and human development chapter. I hope you have a good time studying for the test. Don't forget that you also need to have a quick review Just a brief review on homeostasis as well as human systems and tissues and cell types, and also DNA replication, DNA transcription and translation, and protein synthesis. Reason? Because they're all related, right? The idea of all these things are related to proteins, different cells, DNA. Eventually, it all results in making a baby. I guess that's all. I guess it's long enough as well. I will see you tomorrow and Wednesday. Hope you guys have a good Memorial holiday. Bye-bye.